بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. محاضرة أولية بعنوان Genome Editing can be employed in Bioremediation. إعداد طالبة الدكتوراه أنوار عبد الوهاب مكي، وإشراف كل من الأستاذ الدكتور أسعد محمد رضا الطائي والأستاذ الدكتور زينة وحيد عطوان. Definition of Bioremediation. Bioremediation is a process that uses microorganisms or their enzymes to promote degradation and or removal of contaminants from the environment. The use of microbial metabolic ability for degradation, removal of environmental pollutants provide an economic and safe alternative compared to other physiochemical methodologies. This figure shows the natural role of microorganisms in bioremediation. However, although highly diverse and specialized microbial communities present in the environment do efficiently remove many pollutants, this process is usually quite slow, which leads to poor pollutant of accumulation, which can potentially be hazards to the environment. What are genetic engineered microorganisms? Genetically engineered microorganisms refers to microorganisms like bacteria or fungi, including yeast, that have been modified by humans through molecular biology in vitro, such as insertion of genes in microorganisms or editing their own genes. Steps of transferring gene to new organisms. Target cell with desired gene. Enter to host cell with multiple copies of cloned gene. That result, genetic modified organisms can be used to enhance bioremediation for many pollutants such as organic compound, heavy metals, oil and components, pesticide and radioactive compound. Types of bacteria and pollutants in environment. As the bioremediation is a mechanism that facilitates degradation and or elimination of pollutants from the environment using microorganisms or their enzymes, so genetic engineered microorganisms can be used effectively for bioremedial purposes too. The capacity for degradation of environmental contaminants is high in bacteria. Bacterial strains are capable of degrading various contaminants, example nitroaromatic, chloroaromatics, polycyclic aromatic, biphenyls, polychlorinated biphenyls, and oil components have been isolated for the eventual utilization of their metabolic capacity for bioremediation of polluted sites. Genome editing. Genome editing deliberately altering the structural or functional characteristics of organisms at RNA or DNA level. These organisms include humans and animals, tissue and cells in culture, and plant, bacteria, and viruses. The technique can alter the characteristic of many kinds from the color or number of blooms in flowering plant to some disease traits in animals and plants. The figure shows application of genome editing in different fields: medicine, biology, and biotechnology. Are genome eligible for alteration and how? Genomes are naturally susceptible to alteration, and errors occur every time a cell copies its DNA. If these errors are not corrected by the cell, cancer or some other pathology may arise, or they may confer a competitive advantage, becoming the basis for natural selection. In addition, Genomes may be altered by infection, for example by retroviruses, and ionizing radiation, for example in the case of radiotherapy, X-ray and UV light, which disrupt DNA at locations that may be difficult or impossible to predict. However, these mechanisms are not targeted and so would not be regarded as genome editing. Techniques of genome editing. Recombinant DNA technology. The recombinant DNA technology allowed cutting and then splicing together of DNA molecules. This was developed first in bacteria and their viruses, and subsequently applied to multicellular organisms including plants and vertebrates. Engineered endonucleases. Zinc finger nucleases in 2005 is derived from mammalian transcription factors, are proteins in mammalian cell that bind to DNA and cause a gene to become active. Transcription activator-like effectors nucleases in 2010 is derived from the plant pathogen Xanthomonas species. How do zinc finger nucleases and transactivator-like effector nucleases work? Both are proteins that work in a similar manner, containing one models that can be engineered to recognize a specific DNA sequence and guide. A second attach models to cut the DNA. Although their protein framework differ, zinc finger nucleases and transactivator-like effector nucleases each contain a set of fingers or repeats that can be designed to recognize a selected DNA sequence with a high degree of specificity. Zinc finger nucleases are small proteins, about 20 to 30 amino acids, that are controlled by zinc ion that bind to DNA and recognize 3 base pairs. These motifs were paired with genetically modified restriction enzyme FOCI to make a programmable nuclease that can be recognized target sequence. When two zinc finger models attach to DNA in opposing location with the FOC enzyme in the midst, forming homodimer complex, the zinc finger nucleases are effective and breaks both DNA strands. Transactivator like effector nuclease DNA binding domains with 33 to 35 amino acids repeats motif that recognize each base pair. The difference between zinc finger nuclease and transactivator-like effector nuclease is the number of nucleotides recognized by the protein domains. CRISPR-Cas9. CRISPR-Cas9 was discovered as a part of the bacterial defense against viral attack. Found in the bacterium Streptococcus pyogenes, could be adapted as a programmable system for genome editing. The system components. The system comprises two elements. The first is generically termed clustered regularly interspaced short palindromic repeat CRISPR RNA. The second is CRISPR-associated protein 9 Cas9 in endonuclease. The prototypical CRISPR component from Streptococcus pyogenes comprise two types of RNA molecule that scientists combined into one called single guide RNA or guide RNA. In genome editing, single guide RNA pairs with its predetermined genomic DNA target to form in heteroduplex, so called because the RNA and DNA that pair together are different types of molecule. One region of the single guide RNA matches with its exclusive DNA target site, giving the system specificity, while another region binds to the Cas9 protein. This guides the Cas9 to make a double strand cut at the target site. Comparisons between different editing systems. Characteristic binding principle. Zinc finger nuclease and transactivator like effector nuclease protein DNA. CRISPR-Cas9 RNA DNA. Ease of design. Zinc finger nuclease moderate, transactivator easy, CRISPR-Cas9 very easy. Assembling. Zinc finger nuclease difficult, transactivator easy, CRISPR-Cas9 very easy. Time for construction. Zinc finger nuclease and transactivator about 5 to 7 days, while CRISPR-Cas9 about 1 to 3 days. Costs. Zinc finger high, transactivator moderate, CRISPR low. Efficiency. Zinc finger nuclease variable, transactivator and CRISPR high. Off-target effects. Zinc finger nuclease high but variable, transactivator low, CRISPR high. Single unit or pair. Zinc finger nuclease and transactivator pair, CRISPR-Cas single unit. DNA repair mechanisms. Targeted double-strand genome breaks made by single guide RNA Cas9 are repaired by the ubiquitous non-homologous end-joining or homology-directed repair. Inbuilt cellular repair pathways operating for breaks made by zinc finger nuclease and transactivator like effector nuclease. Non-homologous end-joining. The pathway activation in the absence of donor DNA involve highly efficient. The error-prone direct ligation of DNA ends that causes small insertion or deletions. Meanwhile, activation of homology direct repair requires a donor DNA to initiate homologous recombination between target sequence and donor DNA. Donor DNA can be used to repair a mutation by knocking in. Applications. The use of CRISPR-Cas as a direct antimicrobial tool has been studied recently. Artificial CRISPR arrays have been designed to kill pathogenic bacteria by targeting antibiotic resistant or virulence gene. A recent study used lysogenic phage to introduce a CRISPR-Cas system in E. coli which targets antibiotic resistant genes. The potential impact of CRISPR-Cas9 on industries that rely on bacteria, fungi, and yeast is broad. First, CRISPR-Cas9 is broadening the number of microorganisms that could be used for industrial production. Shaw et al. 2015 investigated the use of CRISPR-Cas9 system in editing the genome of Clostridium celluliticum, a model microorganism for bioenergy research. CRISPR systems can be employed to combat viral disease by disrupting the viral replication mechanism and restoring the infected cell to normality. Cas9 has been used to target conserved regions of hepatitis B virus, which are responsible for virus persistence and replication and are not directly targeted by current antiviral therapies. Ho et al. 2014 eradicated human immunodeficiency virus from human CD4 T-cell by removing parts of its integrated genome using Cas9. Tibas et al. 2014 used zinc finger nuclease. Thank you.